All right, welcome everyone. For those of you that are new to this, I'm Dr. John Lohr. I work in Science Olympiad's national office. If you've been watching any of our other webinars at this point, you're probably sick and tired of seeing me and hearing my voice. Um, but I'm super excited for this one because this is one of our great partners, TKS, who is bringing this topic to you today. And it's one that's becoming I think a really important one for all of us and really important one for science. And we have Esther Kim, who's going to talk to you about AI and using computational models and doing some work in biology, if I'm correct. Did I get that right, Esther? All right. I didn't butcher it too badly, but real quick, just for everybody that's here, this is being recorded. So you guys will get a copy of the video. We're also going to put it up on the YouTube channel, Science Olympiad TV. If you want to ask any questions, I'd ask that you go ahead and use the Q&A feature. It's kind of towards the right-hand side of the Zoom toolbar. It's the box with the question mark in it. The reason I actually put questions in Q&A is it's much easier for Esther and I to track them and figure out, which ones we've answered and all of that. If you have any like technical questions, like, hey, is there a buzzing noise or what's that weird sound in the background? Go ahead and put that kind of stuff in the chat. Also, if Esther asks you anything like, hey, where are you guys from? Or, year in school, that kind of stuff, feel free to put that in the chat as well, because then we get to see that instantly. But after that, without further ado, I'm going to turn over to Esther. Thank you so much for being here tonight.
Thank you so much, John, for having me on. So my name is Esther. I am a director here at TKS. I specifically serve the region of the Bay Area. So I'm actually from San Francisco. I have a few more leading questions to that, but drop in the chat, where are you from? I want to hear where you're located from around the world. See if we have anybody that is actually in San Francisco. So drop in the chat. I want to hear from you. Want to hear from you. All right. All right. Mm. Michigan, what up? Welcome, Michigan. What's going on? All right. We'll get that going. Sanshoshi, so good for you to join us. All right. Colombia, what's up? Illinois. Nice, nice. Man, there's so many people from around the world as well. Well, welcome, welcome, welcome. All right, without further ado, let's get it started. So, TKS is a accelerator for very young people that are both ambitious and curious. And you'll definitely learn a little bit more about our organization later in the presentation. But today, I wanted to tell you a little bit about my background. So, I'm actually a genetic engineer by training. I've spent 10 years working in industrial biomanufacturing yeast for chemicals. But I've also transitioned my role to serve more people and more community because I found that science was very behind closed doors. And it's very hard to access science and practice it in a way that is not at university nor at a job. So I actually worked at a company called the Odin. I don't know if you've ever heard of the Odin, but we sold these DIY genetic engineering kits. So with like a low cost, you can actually purchase some of these parts for as far as a liquid pipette, petri dishes, agar. You can also get some bacteria, and you can also CRISPR genetic engineer anything in your home or garage. So we made these kits incredibly accessible for people to purchase and just learn how to do cutting edge genetic engineering. So I joined this company when it was very early on, and I actually created some really cool and beautiful things. So we actually had this colored bacteria, and I really wanted to see if we could put it on some sort of petri plate and make it visually beautiful because science can also be beautiful as well. So I took an agar plate and I actually colored it with a little bit of charcoal, so it made this jet black petri dish. And what I did with our colorful bacteria, we actually integrated GFP, a jellyfish protein, into E. coli and put some selective media behind it. And I streaked out a plate. So what you see here are tiny little colonies of bacteria that fluoresce under blue light on a petri dish. So I was making kits like this left and right, and I absolutely love the job that I was doing. But it was great because I could package these in really nice kits and then sell them all over the world. So with that, at the Odin, a lot of my work has actually been featured in a Netflix documentary called Unnatural Selection. So I'm kind of low-key famous, yes. If you are very keen to learn a little bit about genetic engineering, the ethics behind genetic engineering, and most importantly, how are people disrupting science in their homes or garages, you can check out Unnatural Selection on Netflix. It's a really awesome documentary, and I'm very biased about it. But with that, we've also displayed our kit at museums, including the San Francisco MoMA, which is the Museum of Modern Art. We've also been featured at the Smithsonian and the Natural History Museum as well. So we've also gotten other engagements, and I was interviewed at Showtime. I've given talks all around the world, including MIT, Harvard, Stanford, and actually last week I did a talk at NYU. But you're not here to yap. You're not here to hear me yap about my like really awesome career in genetic engineering. You're here to learn how to upskill and actually work with AI and validate some of your findings as you go into higher and higher types of academic research. So today I will teach you how to work with AI. So this is incredibly inspiring. This tweet was actually tweeted in April of this year. So actually, I'm sorry, in March of this year, about a month ago, actually. So you could see here this man named Paul. He actually had a dog that was dying of cancer. And this is a very rare cancer. And what he did was he was actually a tech entrepreneur from Sydney. He used he used things like ChatGPT, Grok, AlphaFold to understand the mutation that was causing these tumors in his dog. And he actually cured his dog. He actually made the tumors regress. But this was a very clear example of how you can leverage AI to do research and then actually test it into patients and animals. So they reported, surprisingly, 75 shrinkage in one tumor. So we're really, really excited for, Paul's dog. I hope Paul's dog pulls through at the end of the day. But you can see that even with just AI tools, you can literally build anything and bring anything to life. So where should we begin? A lot of you have been operating in ChatGPT world, and ChatGPT does a pretty okay job, but we both know that ChatGPT also hallucinates, right? It gives us sometimes AI slop, and it's hard to discern what is right and what is wrong, right? So if we were to, expand a little upon like what AI is giving us, maybe we would use Claude. But maybe Claude also is not the right tool to use. So my best and favorite type of research platform to use right now is actually Perplexity. And I'll walk you through what Perplexity gives me versus ChatGPT gives me. Perplexity, I feel like, is a really good place to start. And I want to emphasize the starting point because everything that you generate through LLMs is not true. And you have to validate it somehow, right? You have to get through the AI slop. So when I go into perplexity.ai, I typically give it a very broad question because at the beginning, I just really want to learn about it. I just want to get caught up in what is going on specifically in that industry for that specific disease or cancer. And I just want to be caught up and have foundational knowledge very quickly. So Perplexity helps me kind of parse through the information very quickly. So I ask it, give me the top 10 referenced papers in prostate cancer research in the last 15 years, Right now, I'm obsessed about learning about prostate cancer from the ground up. You have to understand, I am not a scientific researcher for therapeutics nor cancer diseases. So I'm kind of just curious and I wanted to learn more about prostate cancer. So give me the top 10 reference papers in prostate cancer research in the past 15 years. So this is what it comes up with. A listicle of papers, right? Here are the DOI numbers and a brief description of what you can find in some of these papers. So I start with papers. I start with scientific papers. There's a difference between scientific papers and also review papers, review articles. But when you go to any paper, it will specifically say this is a research paper or this is a review. You want to go for the research papers that actually have data, that actually have numbers, that actually can back up and justify what you're actually reading, because this will give you the most amount of information. Now, I know, like when you read your first scientific paper, it's really hard to get through. And I've totally been there, right? I've looked at a paper and I just think, man, this is literally in another language. I don't even know what half of this stuff means. And sometimes it's really hard if we don't have that foundational knowledge that we specifically get in college or beyond in postdocs, that we just don't really understand. But the great part about AI is you can actually ask it very shameless questions, right? AI will not judge you. If you ask a researcher the same questions, they might judge you a little bit, right? So ask AI questions. When I go into a paper, I literally read word for word and I take down notes, and I ask AI questions of, hey, what does an aptamer mean? What does a biosensor mean? What does this mechanical feedback loop mean? And so that way I have a better understanding of what the terms mean and how everything kind of starts to work. And piece by piece, it's kind of like putting a puzzle together. You finally put all the puzzles together and then you see the entire picture as a whole, right? So I encourage you to slog through a couple of research papers because it gets much easier as you go, as you learn some of the terminology, as you learn how cancers or therapeutics or anything kind of works. And it's really great because there's a lot of people in the world. I have to plug Elliot Hirschberg. Elliot Hirschberg is an awesome human being. I actually saw him yesterday at a conference that's in BioBeta. But he writes these beautiful, thought-provoking pieces on how to approach solving the world's hardest problems. And he covers everything from cancer therapeutics all the way to climate tech. But you should definitely check out some of his writings on Substack or Medium. Let me go through some of the greatest scientific LLMs out there right now and tools for genetic engineering or biology. So my favorite above all is ESM3, and you can very clearly see why. So let me just play this little video for you. All right, that is ESM3, y'all. ESM3 is so incredible as a platform. This is a go-to model for protein design, motif scaffolding, inverse folding, and structure prediction. All of these four things has actually taken scientists years to accomplish and do thorough research on to actually publish a paper. ESM can do this, then can crunch this in a single afternoon. So I encourage you to use this model. I dropped in the chat if you're curious on where to find it, but I couldn't even I couldn't encourage you to use ESM3 enough. That's actually one of my favorites. So um, it's multimodal, it's generative. You can clearly see the protein sequence and how it would interact with with other sequences as well. Yeah, it's just beautiful. Here we go. So that's ESM3. The next model I want to share with you is something called Evo2. Evo2 is developed by Arc Institute. It actually has 40 billion parameter genomic language model trained on 9.3 trillion DNA base pairs from across 128,000 genomes from basically every animal and plants, eukaryotes, prokaryotes. It has a nucleotide resolution of 1 million token contacts, and it's truly the frontier model for regulatory genomics, gene expression, whole genome sequence, and you're welcome, it's fully open source. So take a look at this Nature article that I will also drop in the chat as well. Um, that's a really good primer to understand how Evo works and how you can actually play around with it. But I'll also drop the open source code, which is via their GitHub. So that is from Arc Institute. By the way, ESM3 was actually developed by the original scientists at AlphaFold. AlphaFold is also owned by Google. But what am I to do without also mentioning AlphaFold? AlphaFold is also the state-of-the-art prediction model for proteins, DNA, RNA, and small molecules, and their interactions as well. It predicts binding interfaces, critical specifically for drug discovery, really amazing as a gene editing tool, and you can understand hereditary disease, mutations, and you can also design therapeutics based on this protein model. Look, before this protein model, you know what people were doing? People were literally cutting out little pieces of paper, sticking them with tape, and then trying to conform it in 3D space. Like people were building models by hand. But with the help of AlphaFolds, it can actually model and also predict interactions and structures. So super, super cool how far we have come. So those are my top three, which is ESM3, Evo2, and AlphaFold. I encourage you for the next Science Olympiad to use some of these tools to come up with really good proposals and then start developing from there. All right. So learn how to leverage AI to build actual meaningful projects. This is the other half of the calculation, right? It's not enough that we just do research in our corners and then perhaps we share at the science fair. We actually want to launch it in real life. But how do we create real impact from these projects? Maybe you might be in the camp of you're just still figuring out what you want to do in life. And that's okay, right? I'm 36, I don't look like it, but I'm still trying to figure out what I want to do in life. I'm still trying to pursue what is the next big thing that I want to work on. And maybe you're in the same boat too. But you're sitting here and you're like, man, I really want to create an impact. I'm not entirely sure where to start. There's so many cool things happening in the world right now, especially in the biological world, but those things are very hard problems to solve. World hunger, cancer, climate change, all of those things have some sort of biological component that we can actually solve for. But it's very overwhelming to figure out where to start. And at the same time, like I get it, you guys are in high school or younger. You might be very, very anxious about what university you're going to get into or what job you're going to get in the future, right? So you're thinking also about how do I leverage being in Science Olympiad, being in TKS? How do I also leverage some of my varsity sports or being president of the debate club or being president of DECA? You're trying to leverage all of these things while also trying to get into a top university of your choice. But here's the formula, you guys. Follow your curiosity. Okay. So it's not about following your passion anymore because passions can change over time. I'm pretty sure my passions have changed many, many times over my life. But what has always remained true is that my curiosity has always driven me to greater depths and better pastures. So follow your curiosity. And these are some of the topics that we actually cover here at TKS, which is biometrics, blockchain, brain-computer interfaces, cancer, carbon capture, cell agriculture, as well as genetic engineering, my domain. And let me talk to you a little bit about how you can explore your curiosities and perhaps share with you a little bit of the pedagogy that we teach here at TKS. So this is a T, and the top of the T explains the breadth of knowledge. So in my world of doing research in prostate cancer, maybe what I do is I just kind of learn about things. I spend an afternoon, I read a lot of papers, I kind of figure out, okay, this is how it kind of works. And that's what I spend my time in. So I understand this like big, vast ocean, breadth of knowledge, and understand just at the surface level. But then the depth of expertise kicks in. And this is how TKS students develop their T, their breadth of knowledge, as well as they go deep in a specific area. And the way that we've structured it in our program is that for example, if a student is really interested in building in the intersection of VR and AI, what they do is they think that, oh, I really love VR. Let me just explore a little bit about it. Let me read about it. Let me just learn about what people are doing in this domain. But also, I'm also going to do the same for AI. But perhaps like people want to just take that a little bit further. And so they start building their depth of knowledge by building a project in it. And so our focus projects, our focus objective is what we consider a capstone project here at TKS. But like, look, the things that are here on this list is not novel. It's not new. We as TKS didn't invent this. This is actually the engineering process down to the studs. And this is something that I've been using my entire life when I'm building projects from A to Z. So the first thing is just learning about it, learning about how the world works, learning about a specific area, a specific emerging science or technology. The second thing is to basically replicate it. Can I use some of these platforms such as ESM2, Evo3, AlphaFold to just quickly scrub up what that would look like and replicate that? And then the next iteration is replicate it again in some sort of twist. So maybe if you're looking for a new binding site for an enzyme, you could look at the 3D conformational structure and say, okay, I think a protein would actually interact here, and therefore this enzyme would be activated and work, right? And then the last thing is to create something new. So you want to put things into the world. You want to create value out of your research as well. And that's kind of the last part, which is creating. This is our process from 0 to 1, how to build a project really well and ship it into the real world. So let me share with you a couple of projects that I think is really awesome. All right. Um, we talk a little bit about doing the research up front, building in a specific area or a specific niche through this process, which is learn, replicate, and create. But how do we actually launch it into the real world? Well, once your project is somewhat formed and you're starting to get a little bit more validation with your project, maybe through a coach, maybe through Science Olympiad, maybe through an industry expert, what you could do is you can start reaching out to more people because at the same time you're building technical depth, you're also building your network of people. Like that is the one thing that I actually regret not doing it when I was younger or in high school was building my professional network by showing and demonstrating that I'm actually a legit person. I'm actually building something that people want. So we train a lot of students here on how to send cold outreaches. I don't know if you've ever sent a cold outreach to a professional and gotten a response. It's very challenging. It's almost like a science, and also a little bit of an art to send an email to a person and get a response back. So this student, Aiden, we trained him to ask good questions. So, dear Dr. Xu, I'm a grade 11 student in Vancouver, Canada. I'm very interested in cancer treatment. You can find a recent article I wrote about the mechanism of checkpoint inhibitor therapies and the challenges in their use attached to this email. He actually attached a whole one-pager explaining about his project here at TKS. I have great admiration of your lab's research. I really enjoyed reading your recent articles in modeling NF2 related schwannoma. I'm really excited about your research. If you're open to it, I'd like a quick 15-minute call with you, ask you some questions about your work. Your guidance is invaluable to the exploration of medicine. Sincerely, thank you so much, Aiden. I'm sorry I paraphrase that a little bit, but you can see that crafting an email is both an art and a science in order for you to get responses. And hey, look, once you get responses from a university professor that has been researching this domain for probably their entire life, they will actually give you really good feedback. And so that's how you get validation. People don't understand when you're building something, right? You want to make it useful to the world. And it wouldn't be useful if you built in a vacuum. If you built that, if you built something and no one paid attention or no one knew about it, right? Maybe you're building it completely wrong. But the crazy thing is, we as humans, we don't have different perspectives because we can't obtain those perspectives without actually being in it, right? So the best and easiest way to get more validation, to increase your network, to increase your serendipity, to increase the visibility of your work is to put it out there by asking for validation from professionals in the field. And that's how you do it, right? So Diana did this many, many times. She's failed many, many times. She sent about 100 emails to different professors trying to validate her work. And from that validation, she got a couple of professors to give her some really good advice over time. And in one scenario, she actually finally got a research position working at, or she tried to get a research position working at a lab. Unfortunately, they had a little bit of age restrictions. But then again, she was 14 years old. I'm sure it's a little bit different for her now. But I just really want to highlight from her response here on Slack. She said, I made mini projects to a company. You can't just send emails blank and say, hey, I'm really excited to meet you. You have to show some legitimacy, right? So you can send portfolios of work, GitHubs, projects. You could send papers, just to prove that you're worth 15 minutes of time speaking with, right? But also you can demonstrate it with demos. Welcome to the age of 2026 where vibe coding is available to you. If you don't know what vibe coding is, you can essentially prompt a LLM to build you a demo, a video, a website, a one pager, and you don't have to code. You don't have to know how to code at all. So it will spin up really beautiful examples and beautiful demos of this. But Madison also posted it on Twitter or an X so that she can increase the serendipity and increase the amount of people that's actually looking at our project. And in some cases, people actually reached out to Madison. Hey, your model is super cool. It's ultra relevant to what I'm doing. I'd love to do a collaboration with you. Would you like to come work in our lab? And it truly works. It's not just with Madison, but with every other student here, right? So for example, building a bioprinter from scratch. How does blockchain put food on the table for 10 or 100,000 people? Curing genetic blindness with gene editing. It works, guys. Building projects shipping it and putting it out there, getting industry validation. It all works. Let me show you a really cool demo that my students are working on actually right now. They will be delivering it in the next 24 hours to me. But we are doing something called a moonshot challenge. And they have decided that they wanted to take natural occurring phenomena called biomimicry and try and edit pancreatic ductal cells with prime editing. So take a look at this. They actually spun this up last week. So you can kind of see here, they simulated the LMP formation in the pancreas. They also increase it with 99% pancreas selectivity using vitamin D receptors. They've also depicted cargo loads. Let's see if it'll automatically play. Ah, yep. And then it actually gives a prognosis of a very poor delivery system, cell targeting, administration of this, and then it'll also give a score of how it will be genetically edited. So this is a really cool demo that they've spun up. They have no background in software engineering, but they can now do things like this, like creating a demo or a simulation in a matter of an afternoon or 30 seconds or less. Okay. So super, super cool. Now, getting on to the next part. A lot of you might be a little concerned. I know we're all anxious about getting to like a top university of choice or moving somewhere for this, but I know like what the hardest thing to answer is what are actual university admissions officers looking for? Um, and I'm gonna be very frank with you. If everybody at your school is doing the same thing, Scioli, getting really good grades, 4.0, getting high marks on SATs, getting high marks in your in your testing, in your like district testing, being a president of the debate club, right? So everyone's doing the same thing. But then how do you stand out in a sea of people that's also doing the same thing? Right? It's truly something called these spiky outcomes. Spiky outcomes are things that you can demonstrate when you do an internship. Spiky outcomes are things that you build and you develop over time. Spiky outcomes are projects that you've shipped into the real world and created the real world impact. Those are the people that university admissions are looking for to give full ride scholarships, right? So these are the spiky outcomes. Let me just run through a couple of them. This is Shagan. She's so cute. She developed a object detection algorithm by using a video to identify just like common things like a car, a cell phone. And she's learned a lot through TKS and she shipped a lot of projects here. But after TKS, she actually became an intern for the cell physical modeling team at Tesla over in summer in Palo Alto. This student was very, very interested in RNA splicing and how it works. So he demonstrated a very basic way through YouTube, uploaded it, just kind of explaining how RNA splicing works, and now works for a Nobel laureate. Her name is Jennifer Doudna. She works at UC Berkeley, and now he's a research student joining there as well. There's a lot of uncapped systems, right? So if you have uncapped systems, you can have uncapped outcomes. You can learn about brain-computer interfaces. You can learn about neurotech. You can learn about genetic engineering from the ground up. You have the internet. You have access to all of these amazing tools, including ChatGPT, Claude, and Perplexity. But it's really what you do with it, right? If you understand at this level you are the most successful people in the history of the world. You are the most successful people in the history of the world. The fact that you're tuning in and watching this webinar signifies that you have access to internet. You have access to information. You have access to capital. You have people that believe in you, that want to influence you and give you money to build some of these things. People want to give you opportunities like getting an internship at SpaceX or Jennifer Doudna's lab or UCSF or Stanford, right? There's so many people that want to support the next generation of builders. And the way that you're going to get to a really killer university of your choice and then work at some of these really crazy companies or even start your own, it's completely possible in today's world. So you can see here, a lot of our alumni have worked at Neuralink, was a founding engineer at a company. Stanford is the managing director of Nuclei, which is a really good community to get into. SpaceX, longevity. But you also see on the bottom that people have interned at Google, IBM, Shopify, Walmart, Microsoft, you name it. The top 500 performing companies out there, a TKS student has been always the youngest person to work at. For people that really want to found a really amazing company, Alexander Wu is one of our OG goats that actually exited his company to a big company called Coinbase in the blockchain space. Tecton raises about 100 million from the top investors in the space, A16Z and Sequoia. Synex was actually a moonshot idea that Ben Nashman developed, and he decided to develop very non-invasive glucose monitors as his moonshot idea here at TKS. And he has actually raised over $7 million for his idea. Shagan is also interning at SpaceX, like I said before. Ruani is also working at Stanford's Digital Econ Lab. Alishba is working as a deep learning summit in San Francisco, or she's presenting in a deep learning summit in San Francisco. So all of these outcomes can come true. And with or without TKS, it's really how you want to move forward, how you're going to actually create impact. And for some of us, it might be really tough to work on it alone. So we actually have an incredible community of builders. About 1,000, we take only about 1,000 students from all around the world to come build with us for 10 months. And so we are the world premier accelerator to do that. And especially we believe in young people. I gave up my amazing career as a genetic engineer, an investor, and I gave up my career in climate tech to come work with students full time to actually solve the world's biggest problems. So I lead the charge in doing so with other students. So if you want to join the cause and learn more about how you're actually going to create impact, I suggest applying to TKS. Feel free to take a screenshot of this. I'm also available. You can email me, you can chat with me, talk to me about any kind of situation or perhaps some projects that you wanna launch. I'm happy to support you. This is my full-time job. You can also connect with me on LinkedIn here as well. Really good question. So Santoshi says, I have an 11-year-old who is good at math. Would you suggest anything for him? Looks like TKS is for 13 to 17 years old. Yes, so we actually take 12 years old, 12 students that are 12 years old, turning 13 within the next year. So we have a little loophole for that. So as an 11-year-old, they might have to wait another year. I would suggest, especially if they're good at math, go a little bit more into computational biology. I think there's a lot of room to grow from that, but make it a little bit challenging. So, Santoshi, even if you would like to reach out to me, please reach out to me. I would love to sit down with you and your son and kind of figure out a really good project to kind of move forward with in the summertime, and then perhaps he would be ready for next year to join TKS. All right. All right. So any questions? I'm a very open book. I'm happy to answer anything for you. Yes, I'm happy to put my LinkedIn profile. Let me just stop this share really quickly. There you go. And of course, We are taking applications now, and we will be closing applications at the end of this month. Thank you. A
Yes.
17-year-old can apply, yes, right now.
Yeah. So if you're interested or you see stuff, that's and it's sort of the perfect timing. The other, I think, always cool synergy between like the programming that TKS offers and the kind of things you guys are doing in Science Olympiad is there is a natural overlap, right? Like, you know, you're doing a whole bunch of different things in these different events where the beauty of TKS is you're going to get to pick one of them and really dive into it, or you're going to get to do something really cool. And, in some ways, some of the stuff like Esther was talking about in the beginning, do the synergy, right? So you may be the chem lab kid, but you're also doing designer genes, right? Maybe there is a cool way to blend techniques or blend projects or use TKS to help explore the synergy between those couple of things. I'll always remember years ago I don't I don't think any of you guys would have been at the high school level when we did this, but there was a window where we had an event in called Detector Building, where the kids had to build an electronic environmental sensor for that event. And at the same time, we have water quality in, which was requiring them to build a hydrometer.
Hmm.
And so a student was competing in both and decided that what they really wanted to do was expand on the sensor and build it and use that for for water quality. So something like TKS lets you guys really do that in the real world.
Absolutely. And I actually have a lot of Science Olympiad students that I currently work with. And so at the beginning of the year, we start in September. They say, "Hey, Esther, my intention is I really want to be a good contender for Science Olympiad this year. Can we get started on a project?" And I say, "Yes, let's do it." And then the first day of TKS, they propose a project to me, and then I function more of a coach. So yes, I am the director of TKS San Francisco, but I'm also a personal coach. So if you're kind of struggling, "Oh, Esther, what should I do next? How should I reach out to somebody? How do I build in this space? What do you think is impressive? Could I get some feedback from you?" You have me at your beck and call, and I will answer any call that you have and build it alongside you. So it's a little added bonus that you get a coach throughout until you get to that point where you're finally competing at Science Olympiad. And then a lot of TKS students have won a lot of prizes with the program. So there's a lot of synergies with both of our programs. And honestly, John and I are here at the end of the day because we want to support you and we want to support your curiosity and your learning and your building because one day, we're going to be gone, right? And the next generation, you have to take care of the next generation of humanity. But we need to solve some of these world's hardest problems right now. It's just so urgent. But with with the height of AI tools and genetic engineering, anything can be accomplished today.
Well, yeah, and one last plug I'm going to put in is Esther talked about, and I believe if I was listening right, it was the EM or the ESM model was the one where you were doing a lot of work with the protein structure, correct?
Yep.
Just want to remind you guys, if you're not a graduating senior, right, protein modeling is coming back next year. And so if
Ooh.
you're looking for a tool that may help you and give you a leg up on that event in terms of designing your model, learning about functional elements of proteins, learning about folding, it sounds like the ESM may be a tool you want to think about using next school year.
Yes, I dropped it also in the chat. I'm also an advisor and mentor for iGEM as well. And I see a lot more students competing at iGEM with ESM3 modeling. So I encourage you. I almost think it's a requirement for you to explore that as well.
Yeah, that's the thing, guys. And this is, I think, a cool place where you're at and kind of your education and your careers is like these these tools are being just cranked out at such a great rate and they're evolving so quickly. But like a lot of people haven't figured out or don't have the time to learn how to use them or understand them. And I think you guys are in a beautiful place where you do have some of that freedom and flexibility. And, kind of like I think Esther, if you'd agree with this, like you don't necessarily, don't worry about the right one. Really, it's the right one is whichever one you're using, and then you're going to learn and you're going to evolve. I was laughing when you made your comment about chat in the beginning because for probably the last 2 years, I'm almost exclusively using chat. Like the other ones I've tried, it didn't work. It didn't make sense. I wasn't happy with what I was getting. And now over the last week, I ran into something and I stumbled into Claude. And now it's like, oh, you mean I'm going, okay, I got to change our subscription from chat over to Claude because I'm liking Claude better.
John, I completely understand your struggle. And actually, part of my job, every day I wake up and then I benchmark different platforms to see what is the best performing for that specific use case. So, for example, Perplexity, really great for papers and just preliminary research. But I would go to Claude to do more of the computational stuff, as well as build more of those simulation type things. ChatGPT is a little left in the dust, but tomorrow it can change. So
Yeah.
it's very challenging to kind of keep up with the best working model. Um, so every day I have different instructions.
Yeah. TKS is enrolling and they're looking for kids for programming now. If you're interested, I'd encourage you to check them out. Also, last but not least, if you happen to be one of the teams going to nationals in 2 weeks down and you'll be at USC, please find me. I'm going to be all over the STEM Expo on Friday. I'd love to meet you in person and say hi. So again, thank you very much to all the moms out there. Happy Mother's Day this coming weekend. Kids, make sure you really do treat your moms well this weekend. And thank you again, Esther. And you guys all have a wonderful night.
Thank you very much, Sean. Thank you so much. Best of luck today. Take care.